Bismillahirrahmanirrahim. Bismillahirrahmanirrahim. Assalamualaikum and good morning, everyone. Thank you for joining our CME today with the title Harnessing Gene Editing Technology for Treating Retinal Diseases. My name is Khairul Nizam, and I will be the moderator for today's session. Before we begin, let us watch the IMR corporate video.
The Pathological Institute of Kuala Lumpur was established in 1900 by the Resident General of British Malaya, Sir Frank Retinal. Its aim was to promote the health status of the local population and to explore the great and unknown field of tropical medicine. The institute was renamed as the Institute for Medical Research, or IMR, a year later. Initially, the focus of its research was on beriberi and malaria. Subsequently, its scope expanded into the causes, treatment and prevention of a variety of other tropical diseases such as cholera, smallpox, leprosy, tuberculosis, typhoid, filariasis, dengue, melioidosis, scrub typhus, and nutrient deficiencies. With independence, the IMR became the research arm for the Ministry of Health Malaysia, where research conducted has been aligned with the country's health research priority needs. In more recent times, research at the IMR has progressed to gathering in-depth knowledge of allergy and autoimmune diseases, primary immunodeficiency diseases, and through the development of allergy test kits. Cancer research at the IMR has vigorously looked at diagnostics, prevention and early detection, as well as identification of novel biomarkers and targeted therapy for precision medicine. For this, the Institute has been equipped with a specific pathogen-free or SPF animal facility and biobank. The IMR has developed its expertise in nutritional biochemistry, where there is active involvement in preclinical studies on metabolic syndrome, diabetes, cardiovascular health and endocrine-related diseases. The Institute's iodine test kit is used nationwide for monitoring under the Iodine Deficiency Program. The dawn of the next millennium saw many issues that were related to contaminants and pollutants in the environment and in the workplace and its impact on human health. Thus, in a forward-thinking manner, IMR conducted vulnerability assessment on impacts of climate change on health and health delivery. Malaysia is in a region that is full of biodiversity. The Institute has a unique interactive e-database called GlobinMed, which strives to become a referral source on traditional and complementary medicine, or TNCM. The Institute steadily has been moving towards drug discovery programs. The Center conducts preclinical safety evaluation of herbal extracts under Certified Good Laboratory Practice or GLP conditions, assessing efficacy of herbal products using in vivo as well as in vitro systems, phytochemical analysis, and structure elucidation. The IMR has continued to provide in-depth research related to infectious diseases, especially during outbreaks, and it is the reference center for Zika, dengue, influenza, leptospirosis, and melioidosis. IMR is the leading center conducting Wolbachia research in Malaysia. The Institute has equipped itself with a BSL-3 laboratory as a preparedness plan for any possible future outbreaks. Epidemiological research activities have helped to establish the distribution of diseases, its factors, and how it helps in controlling diseases. The IMR provides specialized diagnostic tests nationwide related to organ and blood stem cells transplants, diagnosis of inherited metabolic diseases, inborn errors of metabolism or IEM, genetic diseases and multiple myeloma. In the international arena, the IMR serves as World Health Organization or WHO regional and collaborating center for infectious and tropical diseases. Under various programs, the IMR has produced a few patented products ranging from insecticides, fungi, dengue, chikungunya, saliva and forensic test kits, mosquito detector and trap, paint, anti-cancer toxin, and many more, from which some of the patented products have been commercialized. The 116 completed years of the Institute's illustrious service continues, with new challenges and higher pursuits as the IMR transforms its biomedical facility to support research both nationally as well as internationally. IMR to be a world-renowned institution for biomedical research.
So before we start, please fill in the attendance link in the YouTube description. If you have any question, kindly submit them using the link in the YouTube description as well. Gene editing is an incredible scientific innovation that allows us to precisely change the DNA of a living organism. One of the most well-known gene editing tools is called CRISPR-Cas9. Imagine you have a pair of molecular scissors that can cut DNA at specific locations. With CRISPR, scientists can accurately target and cut the DNA, allowing them to remove faulty genes, fix errors, or even insert new genes. And why is that important? Well, it opens up the possibility of treating or even curing genetic diseases. For example, if a person has a genetic disorder caused by a faulty gene, gene editing might one day correct the error and eliminate the disease. The potential benefits of gene editing are huge, but it also raises important ethical questions. It's crucial to use this powerful technology responsibly to ensure it benefits humanity in safe and ethical ways. To help us understand this very important topic, we have invited Associate Professor Guei-Sheung, also called Rick, to talk about the sciences behind gene editing and recent discoveries in the field. Rick is a principal investigator and leads the Genetic Engineering Research Unit at the Centre for Eye Research Australia. He received his PhD in biochemistry from Guei-Sheung Medical University and was a postdoc at the O'Brien Institute and the Centre for Eye Research Australia. Rick has significant expertise in gene editing technologies, with over 80 publications, and has secured continuous funding from the NHMRC, which is the Australian Government Primary Medical Research Funding Agency, and other agencies since 2012. We are very fortunate to have Rick with us today. So, Rick, the floor is yours. Over to you, Rick, and thank you.
Thank you. So, Nizam, can you hear me? My voice is clear?
Yes.
Okay, good. So, good morning, everyone. So, my name is Rick Liu. So, I'm a principal investigator at Centre for Eye Research Australia. So, before I begin my talk, I would like to thank for the invitation. So, this gives me the opportunity to be here to share our latest research on gene therapy and gene editing. Of course, this is a big field. When Nizam asked me to give this talk, I was kind of thinking the way how we introduce gene editing as a CRISPR gene editing system. So, and today I will just give you an idea, use a more applicable way to tell you how we apply this technology to develop a gene therapy for the retinal disease. So, you might wonder why all your screen go black. So actually, this is a day-to-day life of the people who are blind. So there's a group of disease called inherited retinal disease. The children will lose their vision since their childhood or teenage. After this period, their life would become like black at all. So lost vision is one of the most fear the issue with the healthy. So what is an inherited retinal disease? It's a group of eye disease that caused by genetic mutation, and this genetic mutation contribute to the different fail of the different cell function like photoreceptor, bipolar ganglion cell, and cause the visual impair and eventually blind. So there is around 300 genes has been found to be associated to the inherited retinal disease. However, until now, there's not many options we can use to manage this group of disease. So, fortunately, in 2017, there's the first gene therapy comes developed by the Professor Jane Bannon. So, Jane Bannon and her team is looking into spend a lot of time, almost 20 decades, to develop a gene therapy. And in the 2017, that's first gene therapy comes to treat one type of the genetic eye disease called LCA. So this product called Luxturna. So Luxturna is a gene therapy that approved by US FDA in 2017 for treating the patient who has a gene mutation called RP65 mutation, so associated to a disease called LCA. So Jean and her team are developing gene therapy harness a virus, a small virus called adeno-associated virus, which is a non-pathogenic virus, and that can be packed the normal copy of RP65 gene, and that can be injected into the patient's eye. So this RP65 gene produces a protein called RP65 protein. That's it responds to produce vitamin A. This gene mutant, so that will be lost. The cell cannot produce vitamin A. That causes the unhealthy RPE cell, and then eventually lost the photoreceptor. So this treatment was very effective to preserve vision, prevent the retinal degeneration in childhood after receive the injection, which is also open a hope to the many like genetic eye disease that we now have the solution for that. So it looks like we now could basically cure genetic eye disease, and that can potentially expand to other genetic disease. However, this is not always the case, because this virus endpoint called AAV are small virus. So there's maximum 3.8 kb capacity to pack the gene. There's many gene size than more than 3.8 kb. For example, in the IRD, there's more than 50% gene are more than 3.8 kb. It means that we can't put this gene into the virus. So, and that we won't be able to efficiently deliver a normal copy of gene in the back of the eye. So why are we excited about Luxturna? We are able to treat the inherited eye disease, but we still worry, how about those of the half of patients without any treatment? So make us thinking, apart from this conventional gene therapy, is there any other way we can use to treat inherited retinal disease? So this comes through the development of the gene A technology, or more recently we call CRISPR gene editing. So CRISPR gene editing was first described by the Professor Jennifer Doudna and Avenue Charpentier. They won the Nobel Prize in Chemistry in 2020. So this technology, this advanced technology, allow us to accurately edit the human DNA, and then modify human DNA as a therapy to treat disease, particular genetic disease. So this is CRISPR system original. We're learning from the bacteria. Is a bacteria immune system. So bacteria has to defend like bacterial virus infection. So basically, this system was inside the bacteria. So when the bacteria had the phage, which is bacterial virus infection, the phage will inject their DNA into the bacterial cell. But this system will be recognized that bacterial phage DNA, and then just form it as a memory immune system. Next time when the bacterial phage inject the DNA again, they will recognize and destroy. So this is how the nature CRISPR. And scientists was purpose this system became a useful biotech, and now also even more further to use to build up a gene therapy for many genetic disease. So how this system works? So this system comprise two major components. One is we call sgRNA. So this sgRNA had the two components. One is called transcrispr RNA. So this is a loop step. So that's used to attract and bind to other components. And they also had another called sgRNA. This sgRNA actually can be complementary to the target DNA and recognize that DNA. And this sgRNA can be hooked by to the called Cas9 protein. Is a molecule scissor. So this molecule scissor able to cut the DNA when this system searching through the genome and recognize the sequence they are complementary. to the CRISPR guide RNA sequence. So after that, so this molecular scissor would be asked to cut the double-stranded DNA via recognize another important sequence called pensi. Pensi is a few nucleotide sequence. This motif allow sit as a security for CRISPR system. Even though this complementary sequence would be recognized by guide RNA, the Cas9 won't be cut this double-stranded DNA unless they also found this pensi near the target sequence, and that will trigger the double-stranded DNA break. And once double-stranded DNA break, that would be induce a two-cell mechanism for repair the DNA. One is called non-homologous end joining, which is an error-prone repair. So this repair system basic is a forming like insertion or deletion to make the DNA join again, which is can use this approach to kind of disrupt the gene, disrupt the DNA, and then silence the gene as well. And also another mechanism called homologous direct repair called HDR, which is most like useful because we can introduce a donor template. Through the homologous recombination, they can recombinate to change their sequence to what we interest, which is a concept of use this system to do a gene correction to treat the disease. But unfortunately, non-homologous end joining was in the somatic cell. The non-homologous end joining was around 98 to 99%. Only 1% with the HDR, which could be the issue to use a HDR for treat the disease. So in 2014, so our group first to get to thinking, yes, this system can be useful for treated retinal disease, but how we deliver this system into the eye. So our group was the first to use the AAV, which is virus being described, used in the clinical to do a conventional gene therapy. And then we are able to fit CRISPR system into a single one AAV, and then we feed the guide RNA to another AAV, create this called dual AAV system. And then so we inject that dual AAV system into the mouse retina. We decide to target the green fluorescence, and then we employ a mouse model. They had the green fluorescence expressed in the mouse ganglia cell and mouse retina cell. So, and after injection for 4 weeks, we clearly see that system specifically recognizes the green fluorescence GFP gene and then disrupt the gene, which is by the non-homologous end joining mechanism, and that reached to 80% of editing in the retina. So this was first experiment demonstrate we can use a virus to carry that efficiency to target retina cell and edit retina cell. It was quite exciting to move to this. However, as I saying, the concept of doing the gene editing, we wanted to correctly to repair the mutation. And unfortunately, so even we can successfully deliver the system into retina, but we only can achieve this called destruction, not able to do the repair. So later on, that several group from the particular David Liu's group from the Boyd Institute and Harvard University. So they further use this system to be more applicable by the different way to repair the mutation. For example, the team first developed a code-based editing. So they use a code dead Cas9. So they remove the one of the function, the Cas9, to cut the DNA. And then the fusion with the effector called deaminase. This deaminase is able to change just single DNA base to another base by deaminase. By this deaminase, one of the structure of the amino acid, and then that can be lead to be get the DNA repair and then repair from one codon to another codon, and this technology will now allow us to fix the single point mutation along with this a DNA repair system. And also, apart from that, we also the group also later on also developed another system called prime editing. So this prime editing is instead of use the deaminase, they use a reverse transcriptase. Is another enzyme that enzyme able to do the DNA repair and then incorporate the long sequence. So it means that now we, apart from do the single base conversion, so we also can use this prime editing to replace any sequence that we interest to be repair the genetic mutation and reach to the effect of the therapy. So this was quite exciting to more and more this type of technology coming from and help us to address disease. So now I give, and you might wonder how far this technology been so far been gone to a clinical status. So it actually is quite fast development in the last few years. So for example, this is at first use CRISPR gene editing technology to treat the inherited eye disease called LCA10. So the system is employ a CRISPR non-homologous endogenous repair mechanism. So they create a virus, dual virus, carry the CRISPR system and then two guide RNA. That two guide RNA able to locate the CRISPR Cas9 into a deep intron area, where this deep intron area has mutation that induce the incorrect splice, so create this not the functional protein or abnormal protein. So in this case, they're able to put the two CRISPR enzyme and guide RNA to be to near the mutation site and then cut off the mutation region, and then and then that helps to be the gene splice normally, RNA splice normally, and making the normal protein again to restore the CEP290 protein. So that helped to be prevent the vision loss. And this trial was quite success in terms of stop the getting to prevent the visual degeneration in progress in several patients. Also, this CRISPR system also been used to modify the cell, and that use an ex vivo modification and then transplant back to the patient as a cell therapy. In this case, so this is the first CRISPR therapy been approved by US FDA in the patient in the December 2023 last year. So they use a similar system with the CRISPR system to disrupt the gene called BCL11.1. So in the patient who has the thalassemia or sickle cell disease, so they often had the mutation with their beta chain of the hemoglobin. So that's lost. So because the hemoglobin cannot carry the oxygen. So in this case, and then in the normal fetal condition, so cell would be produce a called hemoglobin F, which is coupled with alpha chain and gamma chain. This would, but however, this fetal hemoglobin will be decreased after birth. So, and that's because this gene called BCL11 translate as a protein to trigger that transition. So, and then the group, they use a CRISPR system to disrupt this gene, so there would be no BCL11A disease. So that makes that fetal hemoglobin F can be produced, and that it's instead of could be replace the faulty hemoglobin A, and that function make the function of the oxygen in terms of in the systemic. And also there's another case which is use CRISPR to disrupt a gene that leads to the abnormal folding protein and cause in the metabolic disease. And this is also a really significant case and been gone through the clinical trial. So overall, there's a many trial being proposed and even reached to phase 1 and some of phase 2 and phase 3 and get one approved. As the case of the sickle cell disease and the thalassemia. So they also demonstrate the useful of a CRISPR system in terms of the working in terms of the cure genetic disease. Of course, there's still a lot. Some of the trials are ongoing. Use other CRISPR systems like base editing and prime editing. So we are looking very exciting to see a result from their progress into the different phase of research. So again, come back to our previous question. Now, we should say we had a very good tool to like conventional gene therapy and CRISPR gene therapy. But is that at all? The answer is no, because we use a CRISPR system to target the cell, to target the genome DNA, and that's also a concern with code of target. If we are correct one gene, but other gene also been added or other region been added and cause a problem, so that would be like you cannot reverse this effect because we are added DNA. So the reason the CRISPR-Cas system is really specific, really precisely edit the DNA is because the pen site I just told you earlier about the CRISPR system. This pen site acting as a kind of the security guy. So this is a security guy help to actually maintain the sort of the security, not allow CRISPR-Cas9 to edit many locals. And so, however, this security guy also acting as an issue to prevent this therapy development, because not every mutation, not every mutation gene had the pen site nearby. So which makes it very difficult to use this system to treat every condition. So that's why. So later on, the scientists also explore other methods to be able to do better or to address the gene that cannot be treated by CRISPR-Cas9 system. So people start thinking, if we can't do the CRISPR-Cas9, can we use that target RNA? Can we target RNA, which may be safer because we are not touch the DNA, we only edit the RNA, which is safer for now. And also, so actually, the CRISPR system also can be found to be, there's another class called Cas13. This class of the RNA and be used to target CRISPR RNA system, and basically is similar to CRISPR DNA editing. So we had this called CRISPR-Cas13 enzyme is also a molecular seizure, but different to the Cas9 is this molecular seizure is targeting the RNA, not the DNA. And then this system also can be guided by a guide RNA to search through the transcriptome and then found the complementary RNA and degrade RNA. That's where the system works. And then, so our group was very interested in this system is because we've been looking into the DNA editing for many years, but we found there's a limitation in terms of editing site or editing efficiency for treating a genetic eye disease. So this Cas13 RNA editor might be a way to go. And then for most of the next few slides, I will tell you about the two cases of we use CRISPR-Cas13 RNA editor to treat the eye disease. But most of it will refer to Cas13X because it is a smaller Cas13 enzyme that we're able to fit into a single AAV system for treat the disease. So since it's targeting our RNA, so we can use this system to basically bind and target the messenger RNA, which is, for example, a a gene encoding a protein that contributes to pathologic feature. So we can use this CRISPR-Cas13 to recognize that RNA degrade. So know the pathologic protein will produce, reach a therapeutic effect. And we can also learning from the DNA editor. So we can add in the effector to the inactivate Cas13 to be able to edit the RNA transcript, for example, make a single base conversion, so that able to translate the normal protein and maintain the biological function. And also, since RNA editing is modified RNA, so we can also use this system to fusion with different functional modulate effector to modulate the RNA function, like RNA to mediate the gene translation and splicing. So to use this mechanism to address different type of disease. So the first, I will tell you a story in our lab. We use this system to target a gene called VEGF to treat neovascular eye disease. So neovascular eye disease is a disease that often is seen the abnormal growth of leaky blood vessels in the back of your eye. So often you see in the disease like age-related macular degeneration and diabetic retinopathy. And scientists have been found that this abnormal growth blood vessel is associated with a protein called VEGF, is a vascular growth factor. So anti-VEGF medicine has been quite efficient to use to manage this neovascular eye disease. However, the current treatment requires frequent inject this protein constantly for every 4 to 12 weeks for many years, even lifelong, which is cause a burden to patient and clinician and whole healthy care system. So our idea is that if we can purpose this CRISPR RNA editing system to degrade the messenger RNA of VEGF, and then we might be able to create a new gene therapy that able to manage this type of disease, and it can be used to and so under this concept, so we are design a CRISPR-Cas13 RNA editing system, so to target the VEGF messenger RNA, and then apart from looking for the targeting efficiency, we also wanted to explore a bit further to look at whether how specific these systems are. So, and then we will be looking into the corrected cleavage, which is once you activate the CRISPR enzyme, so it will be cleavage the messenger RNA of VEGF, but it also potentially cleavage any RNA around this if present in nearby the CRISPR-Cas13 enzyme. And also we were looking for the homology of the guide RNA. See, apart from target the VEGF, whether there's another the high homology sequence can also be targeted by this system. This is called off-target cleavage. So we create a construct that and test in the cell. And then so we compare to a shRNA quite common use in the past, and also another Cas13 system called Cas-RX. But this Cas-RX is a big, the enzyme is quite big, so it's very hard to fit into AAV. So produce like quite good titer of virus, and so in this case we are transfect this vector into a cell, and we found both the Cas13 BT3, which is the one we interest, and also the other version of Cas13 are very effective to reduce VEGF messenger RNA expression and outperform than the shRNA, and both are able to reduce protein as well. So first we look into the corrector cleavage, and in this case we are performing an RNA-seq to compare to the kind of the CRISPR-Cas13 BT3 harbor with the VEGF sgRNA, which is only target VEGF, compared to the scramble sgRNA control with the Cas13 enzyme and Cas13 enzyme itself. And the data we found this system was quite specific. We only found a few less than 100 other RNA being targeted. In particular, there's there's no downregulated gene being found. So looks this system quite specific. And then also we look at for off target. So we performing some of the bioinformatics to look at the sequence homology to the region of the VEGF that we designed to target. So and then look at this gene. So we didn't see any of these off target with this top 10 gene, and this also demonstrate this CRISPR RNA editing system to target VEGF quite specific. And we also done some experiment in the organoids, the right retina cell, so to demonstrate it also work in the retina cell as well. So next we are moved. So after the in vitro and ex vivo, we now want to see if this can work in vivo in the animal model. Since this system is quite specific, only target human. We design only target human VEGF. So we employ a mouse model called Kimba mice model. This Kimba mice is a transgenic mice express human VEGF in the retina, and that human VEGF also triggers the vascular leakage and vascular abnormal in the retina, serve as a really good model for us to test the RNA editing anti-VEGF therapy. So we pack this CRISPR RNA editing system into a AAV, and along with the another virus that carry a mCherry gene as a reporter. So we could through this reporter know how good the virus infect, where they infect. So and then we inject the virus into the 6-week-old animal, and then look at 8 weeks. We look at the live image, and we take the RNA to do a qPCR to look at whether how the efficient the messenger RNA been knocked down. And so you could appreciate that in the control or non-targeting the case. So you will see a lot like vascular leakage happens here, and then also but however in the group received the VEGF targeting, you see the leakage almost no in the mouse retina, which is demonstrate that RNA editing therapy are quite efficient to control the leaky blood vessel growing. And then we take the retina to look at the gene expression of human VEGF-A and mouse VEGF-A, and we found this is only specific target human VEGF, not the mouse. Again, demonstrate how specific this system, because mouse and human only have one nucleotide different. So that's quite exciting for us with this data. And you might wonder how long this can be translated to clinical. Actually, 2 days after our PNAS paper published, so there's a Chinese gene therapy company called Gene announced their first FDA clearance for human trial with their gene therapy called HG202. So this HG202 is exactly same like CRISPR-Cas13 RNA targeting system for treat neovascular AMD with the targeting the VEGF. So, and we are quite excited to see this trial coming, because so in the next few years we will know this type RNA CRISPR RNA targeting therapy will use for managing eye disease or not. So so since I don't have much time, so I will quick tell you another application of use CRISPR Cas13 RNA targeting system to treat inherited retinal disease. So similar to the DNA editing. So what we usually do is we use CRISPR system to edit or correct or modify the gene in the genome, so that can lead to produce normal RNA and the protein as a therapy. But for CRISPR-Cas RNA editing system, we not targeting any DNA, so which is can consider safe, because once anything goes wrong with the DNA, genomic DNA, you can't go back. So instead of target genomic DNA, we target RNA, and that can also produce a protein. Although we can't produce AAV normal protein like as a normal is, but a lot indication showing that if you reverse like 20 to 50% of protein, that able to actually maintain the cellular function. So in this case, so we create a called base editing, RNA-based editor. So this base editor is allowed to correct a single mutation, like I mentioned earlier, a DNA-based editor. So the reason we wanted to develop this is that when we look at the spectrum of the mutation along the retina, inherited retinal disease, we found there's more than 70, there's around 72% are point mutation. It means that if we can develop a therapy that able to just do a point, single point mutation correction, that we can address quite substantial number of the IRD patient. And how we do that? So we could fusion the RNA-based RNA, CRISPR RNA Cas13 with the amino acid called ADA or APOBAC. So this can help to do a single base conversion in the RNA codon, so achieve to the therapeutic effect. So what we decide would be that we use a CRISPR Cas13 BT3, so and then fusion with ADA as a proof of concept. So this ADA can do the A to I can be subsequently read as a G, and then produce a normal transcript and then translate to a normal protein to maintain the visual function. And then in this design, so we could design the guide RNA that's guide to this CRISPR RNA-based editor to the region that we want to correct. And then in particular in the correct site, so we will do it called AC mismatch. It tells the system that this AC mismatch is the most important site. You have to make that correction, become A to I to G. So we first to do a proof of concept study in the disease model called RP65. So we construct this RNA-based editing system and then design the RNA to target the nonsense mutation in RP65 gene, which is happen in the patient with the LCA. And then after that, so we not only this target one mutation, we are targeting the 6 mutation, which is pathological mutation we source from the clinical data. And this also what we wanted to see whether this system, this CRISPR RNA-based editing, how efficient can they correct all region of mutation, or is it something else? And our data showing that introduce this system in vitro in the cell, so we are able to correct like 992, 993, and 1205 mutation, which is reached to about 60%. Yeah, and then however, there's a 3 others, either no editing or low editing. So this also tell us good news that we could edit about half, but bad news that there's some factor actually stop the CRISPR RNA-based editing to bind to do the job. So and now there's important work for us to figure out what is the factor, perhaps the RNA structure or number of RNA binding protein or any else other factor. So this would help us to develop a better RNA-based editor to address all the mutation across to a disease, different disease gene. So and I think RP65 is not is a good proof concept, but real money is used to treat the disease that too big cannot be fit into the single AAV vector. So this is most likely more significant. So and in this case, we are working into a disease called Usher syndrome. This Usher syndrome is a genetic disease caused by a group of gene mutation. So the kid was born with the hearing loss, and then they gradually lost their vision from the 10 to teenage, which is very devastating disease happen in the kids. And unfortunately, one of the most the gene called USH2A, which been shown to cause the Usher syndrome, are very big, almost almost reach to like 20 kV like 20 kb, so which is impossible to do a traditional gene therapy. So, and then for this, we designed a CRISPR RNA-based editor to target the human and mouse. And then we showing that in mouse, we reach about 50% half. And then in human, we can reach about like 30 to 35% editing efficiency, which is quite good and compatible. So then we move to animal model. So we pack this CRISPR-Cas13 base editor into a AAV vector, and similar, we co-inject with the reporter to see the delivery efficiency. And then we create a mouse model. That mouse model harbor with the same human mutation. Unfortunately, the model doesn't have phenotype because the human and mouse protein slightly had different function, but at least this will allow us to demonstrate proof concept, demonstrate the genotype in terms of the correction of RNA to reverse a mutation to produce normal protein. And after we inject in the 10-12-week-old animal, and then we was doing the live imaging and sacrifice in like 8 weeks after. So here is the image that 8 weeks after treatment with the quite good fluorescence indicator the virus delivery are quite efficient in terms of retinal cell. So after 8-week treatment, we take in the RNA. So we found that's good editing about the 27%. And there's no other bystander effect of target being found. And more important, it doesn't impact the retinal function by some ophthalmic assessment. So which is also exciting for us. We're thinking this is a good tool, and then we can reverse the RNA mutation and also not damaging the retina. So having tell you a lot this the DNA editing and RNA editing via CRISPR system, I think overall we now have a really good tool to address different type of mutation. So in my mind, in the future, what this genetic medicine looks like is that once we identify the mutation from the patients, we can allow us to knowing like which the therapy is the best for them to treat this type of disease, and then we can use we can take the patient's cell and test in vitro, and then to confirm the way it works, and then we can make in sort of the therapy outside to do a gene therapy in real patient's eye. And of course, I also thinking that each of the technology developed from the CRISPR system are very, very useful. So because once we develop more different tool, it means that we had a lot different technology and tool inside. So this technology can help us to address different type of mutation or different type of condition. For example, some mutation it's better to use DNA editing. Some of mutation better use RNA editing. Some of mutation even you don't need to do editing. You just need to use a CRISPR system to activate or suppress a gene. So which is reflecting as how technology development and eventually can benefit to the disease that we never able to treat in past. So overall, I think that's all my talk today. But I wanted to make the acknowledgement to all my team at CIRA, Center for Eye Research Australia, and Uni Melbourne. Without them, I can't do much about all this work. These are all hard work. And then also our collaborator working on stem cell, in vivo model, and also we're in UTEST. And so we initiate a lot of these projects I initiate when I start my position in UTEST and then move to Center for Eye Research Australia. And also thanks for all the funding support. Without this money, we can't do anything. So thanks for your attention. So I'm happy to take a question.
Thank you, Rick, so much. It was an awesome science and talk that you delivered. I think you've given us a great overview of the field, starting from gene replacement therapy pioneered by Jean Bennett to Jennifer Doudna's breakthrough of CRISPR-Cas9 and its evolution to base editing and prime editing. And finally, we are now in the age of RNA editing, which is, I think one of your work here is showing good promise to be used in clinical translation. So I think we do have time for a couple of questions. I think I start off with the first one, Rick, which is when we think about CRISPR-Cas9 as a treatment, right? Because it goes to the DNA source and then does correction where the mutation is originating from. But with RNA editing, you're actually editing the RNA, which is the product of the DNA. So in terms of delivery, how often do you imagine this treatment will have to be given? Like for CRISPR, it has a one-off treatment, but how about for RNA editing? Does it have to be given periodically? So how do you imagine this treatment in a real clinical setting later?
This is a very good question. Like this is also distinguished from the DNA editing and RNA editing. So of course, DNA editing is considered one of editing. So it's an optimal. I think this is the best treatment for patient. But unfortunately, there's a lot of limitation with DNA editing. For example, you require pen site nearby. And then so current editing technology, I think are pretty confident. But this is also beginning. You don't know what happened in 10 or 20 years after, if that's an issue. And the RNA editing, I think, is give another tool to the patient who the mutation, you don't have any pen site nearby, you can't do any DNA editing. So, but this RNA editing, of course, you need to constantly express a CRISPR component and edit the mutation. So so that's why we require use AAV. Lucky the CRISPR-Cas13 is quite small. We can fit into AAV and then deliver in the retina. So from the current clinical trial with the conventional gene therapy, that now is a 10-year mark. So this 10-year mark, so from clinical study, hasn't seen any significant decrease of the visual function from the clinical trial. So it means that use AAV to deliver is able to maintain the gene expression for very long. And then now several groups are ready to look into if one day if the therapeutic decline, we might be able to administration of the AAV vector, so that can keep the therapy continue going on. So I think that's both therapeutic use CRISPR system, RNA DNA has their own issue and their own advantage. It really depends how we use it.
Oh, thanks, Rick, for the answer.
Should I stop sharing screen or?
I think you can leave it on. Yeah. So the second question, which is something much more relevant to our region here in Southeast Asia, which is thalassemia. So someone is asking, how can we decide which method to use to edit point mutations in thalassemia? Is it CRISPR-Cas9 or CRISPR-Cas13, or do you have any other suggestion?
So in this case, I would be fully standardized with CRISPR DNA editing with Cas9. Right. So of course the thalassemia they can be the many different types of thalassemia, but particularly significant where the treatment has been approved by FDA, which is you modify the CD34 cell by a knock down a gene, which is use CRISPR-Cas9 to knock down a gene. In this case, you just pick up a region that easy to knock down. So scientists have been optimized to knock down that specific gene and then allow the fetal, the hemoglobin can be produced to continue produce to supplement the actually function of the hematopoietin cell. So in this concept, so because we don't require any like repair, require any correction, just gene destruction, and we know that CRISPR-Cas9 is good job to do a gene destruction. So in this case, I think the ones of the DNA editing would be the goal. And then so it again, I think I wanted to stress this concept that that in all this CRISPR system, I don't think any specific system is better than another. It needs to depend on what the disease condition, what the mutation, and choose the best approach as a treatment. treat the patient and manage the disease.
Awesome. Thanks, Rick. Perhaps we'll just take one last question. I think this is more clinical-oriented question, because I think you showed in your presentation that there are a number of clinical trials based on gene editing are coming to the clinic, right? So I think in Malaysia, we also receive invitations to be part of some of those trials. So some of the groups are asking, so how sustainable is CRISPR-based treatment for long-term effectiveness? And how long should patient be followed up to see if there's any adverse reactions, or basically to establish a safety profile of the treatment? So this basically we can ask for your opinion on this, Rick.
Okay, so overall, from the table, you see there's many of the CRISPR system, many like CRISPR-based gene therapies been developed to manage different disease, start from eye disease, metabolic disease, blood disease, and cancer. But it really depends on what type of the disease and outcome you're looking into. For example, for a lot genetic disease, so you wanted to use a system that able to efficient to reduce the pathologic gene expression, and or you wanted to make the gene restore the gene function again. So for the primary, for example in the eye, so currently there's for most of these clinical trials, we didn't see much called the side effect, but that's still too short. It need to be long term, and also you need to look at it to the how the data showing from the preclinical study. Most of these all showing the safe, but in terms of the how to evaluate safe, how it does, I don't have a good answer for this, because it's a disease center disease. Different disease, they have the different opinion, and but I think my suggestion is that if this is good treatment and there's no better management way to do that, I think I fully support to try it. And but however, need to consider as a outcome of the treatment, if and then they can make the balance in the future. So in simple ways, I don't have answer for this, but I think time will tell us. And also I think we should keep hope for this technology, because it really changed the way to manage the disease that we cannot actually treat in the past.
Yeah, we really appreciate your advice and insights into this, Rick. I'm afraid this is all the time we have for today's seminar, but I'm sure Rick is more than happy to take more questions, so please do get in touch. And also, I think Rick is also looking for potential collaboration from us here in Malaysia. So please also get in touch if you're interested to, work on some research collaboration. And if you're looking for a PhD supervisor, I would recommend Rick for you. So please get in touch as well. So because I know I'm one of his students. So before I end, firstly, thank you, Rick, so much for coming on the talk for today. We really appreciate it. And thank you also to Dr. Jelina from I'm Genetic, Cik Effendi, Cik Rizal, Mr. Andy, and the whole team for today. Hope we're going to see you in the next round of talks in IMR. Until we meet again, thank you.